Hello, everyone. Welcome to the 20th episode of eTalks, where we're joined with Mariam Chichik as our moderator and Brinda Guntur and Or Doni as our guest speakers. Today, we'll be talking about research as a learning journey through the lens of Brinda Guntur and Or Doni. So Mariam Chichik has been an active member of Argonne Art and Academy since 2022. She's going into her last year at Rutgers University, majoring in cognitive science and minoring in math. Additionally, she is passionate about music and hopes to improve in this area. And currently, she plays the oud and the guitar. Or Doni, our guest speaker, is an undergraduate student at the Rutgers of University and also a researcher aspiring to work at the intersection of clinical medicine and scientific research. And Brinda Guntur, our second guest speaker for today, is a cell biology and neuroscience and computer science student at Rutgers and also works as a research assistant at the Center of Advanced Biotechnology and Medicine with Mariam and Or. Besides research, she's passionate about patient care and has experience as a medical assistant and EMT. And in her free time, Britna, enjoys arts and hiking. So Mariam Chichek, I'm handing the microphone over to you. We are all excited to hear from Brinda and Or about their research experience and what they've learned so far.
Hello. To start off, I want to mention how I got to know Brinda and Or. So we were in this year and last year I was in a SURE program at CABIM, the Center of Advanced Biotechnology and Medicine. So it holds a summer undergraduate research program. And this year, Brinda and Or were in the same program. So we would meet weekly and we all work in different labs in the same building, and we met weekly to work on our presentation of our research for the end of the summer. And that's how I got to know them. And what inspired me to invite them here was their passionate conversations about research and their classes, like how how they were giving each other comments on classes to take, why to take those classes, and in general, I think they're very interested in neuroscience and bi biological sciences overall. So I thought maybe other people would benefit from listening to their conversations as well. So this will be learning experience. This vibe will be hopefully a learning experience for us all. Um, you Husna already mentioned about you guys, but is there anything you would like to add to what Husna said about you? What are you? What do you like? What are we talking about today?
I think we'll get into, I guess, because we're going to talk about what we're doing for research. But I think the main thing that I would tell someone, I don't know, when I came into Rutgers and in general, people say, oh, research is X, Y, and Z. It seems really scary. Um, it is a lot. And it's not like anyone is good at it or is born good at it. But I think it's like the reason we're talking about it right now is it's important for everyone to have an understanding of what research is and sort of how it impacts our daily lives. And beyond it, if you find these things interesting, then looking for a way to get involved in research projects, whether it's at an actual university or civilian scientist projects, which there's many of. So we're trying to sort of introduce people to what research is, and then, it's good general information, and it's also something you can get involved with if that's your cup of tea.
So let's
So, and like
Yeah,
Or said, it's true. Like every single one of us has things and we're always curious about things. So just because you're curious about it and maybe you want to turn it into a passion, this could be a really nice podcast for you to listen to. And even if not, maybe you'll learn new things and maybe you'll find interest in other things. So it's always a good experience. Just yeah.
So based off what you guys said, what is research?
okay. What is research? I think I can start. And so what is research? And in the most basic sense, research is a way we have of asking questions. And we're always asking questions as people, whether it's like, I don't know, what do you want for breakfast, or what's the meaning of life? We have a bunch of questions and we have a lot of different tools for answering them. So you can see ice cream here at the bottom of the screen. So one way I like to explain it to people is like, if I ask you, Miriam, what is your favorite ice cream flavor?
Blackberry.
Okay, blackberry, awesome. Was that research? Not really. That was just me asking you a question. I wanted to know. But then if I ask you a question, or as Rutgers students, I want to find out what the most beloved flavor of ice cream is at all of Rutgers, and find it in a way that's reliable, then that's research. And so that's sort of the difference. And maybe the most beloved flavor at Rutgers is blackberry. Maybe it is something completely different. So all research is it's a way of using different tools and methods to get answers we're very confident in, or as confident as we can be in, to these questions that are more complicated and more sort of evidence-based. So if I wanted to figure out what the most beloved flavor of ice cream is at Rutgers, there's a bunch of ways I could go about it. Maybe I would do a survey, maybe I would partner with ice cream shops around. And then I would have to do all of these different statistical tests to see if my results are meaningful. Maybe it's a tie, maybe it's so close that you can't really tell, and you have to talk about multiple flavors. So it gets boring very quickly when you get into these details, but that's sort of the idea of research. And then Brinda, if you want to continue for the validation publication.
Yeah, absolutely. Just like Or said, research is about asking questions, but just because we collect data doesn't mean that it's completely valid right at the moment. There could be errors or outliers, and so it's always important to always redo the research of multiple times in order to ensure that the results are similar every single time in order to actually verify a fact or actually state something and validate it. So, after you're done with asking a question and doing a whole experiment, it's always nice to just do it again multiple times. And once you get the results the same amount, or you validate it, you can actually put in publications and review papers, which is how like most scientists learn about new things. So it's a really cool process. Just another important thing to note is like population size really does matter. So like Or said, like when you're like finding surveys or trying to ask other people, the more people you ask, the more closer you'll get to better or more accurate results. And of course, it's not always the easiest thing, but it's just important to note that that is definitely a key factor in research.
And then also, oh, sorry, go on.
I would also like to add something. Maybe it's very good to ask good questions. And a very important thing I learned, I guess, throughout the process is sometimes like after asking the questions and seeing the results, you gotta also objectively observe what you've seen without like thinking about what you assumed or asked in the first place. So like paying attention to the detail in that sense is very important as well.
You're right.
Right.
You shouldn't just there is one thing I want to add to that, because sometimes manipulations and biases can add to that. So just because you want a hypothesis to truly work out, don't try to get your emotions in the way, because as scientists, you shouldn't let emotions get in the way. So that is a really key note to have, but it's always really nice to see your results play out like the way you envision. So yeah.
That can be an issue, especially if you get into research because you really like something, you want to prove it works. Then it becomes a sort of bias where you have to be aware of it and sort of let it go. Like the experiment I'm running right now, we have a hypothesis where we think something works in a certain way. And if I get any result that shows that it did work that way, that's awesome in a sense, because it's allowing us to tell a story where, look, this thing works in this way that we thought would happen. But then I'm also getting some results where that's not the case. And so I have to repeat my experiment and I have to show these results. And so some things that can happen that are manipulations are where people will sort of avoid telling the whole story or only tell the parts that worked, which is a big issue in science. So that's something to think about. Also, when you're reading an article or the label of a product, think about what did they test, what did they not test, and sort of why is this the story I'm being told, what's being left out, what could have also been asked. Yeah, should I move on?
Yes. And so after talking about what research is, now if people are interested, how would people get involved in research?
I can start us off for this one. So basically, it's really nice that you want to make research a career. So the ways you would go about it is don't be afraid to reach out. Honestly, um, a lot of people stray away from reaching out because they think they don't have the adequate grades or knowledge or passion for it, but you never know until you dip your feet into it. Nothing is set in stone, so always try it. It doesn't hurt. And so some ways you can do that is apply to your local program. So it could be that you're a university student and you can ask college campuses, hey, like, what are some programs that you guys offer in order for me to get involved in? So I feel like those are really, really good to dip your feet in. I know at Rutgers, there's Aresti and Sure that are really, really nice. And of course, there could be a lot more. And that's something you can always find out through like offices and departments. So it's always nice to ask. I think another key factor is utilizing email and LinkedIn. I started my research journey back in high school when I was a junior, and the only way I was able to do it was because I cold emailed. And a lot of people are like, well, you might not have the experience or the grades. Like, how do you get into it? Talk about your passion. Be real. It doesn't matter. No one cares about the achievements you have. People care about your attitude, your passion, and basically why you want to do it. Don't have a silly reason. Like, truly be curious about something and go into it. And people will honestly see that ambition and always try to help you out as much as they can. I do want to note that sometimes you might get rejected, and that's okay. Rejection is redirection. You'll always go through something in life, and you just have to like realize that maybe that wasn't meant for you, but another door is there for you. So always utilize email and LinkedIn. Honestly, key factors. And I'll let Or take over.
Yeah, I'll just add that they're being rejected sometimes. It's for me at least, and then maybe most people, it's being rejected almost always. I've emailed many more professors than have responded and then have talked to me and gotten to work together with me. But with some things, you only need one, so really important to just keep trying and not take things personally. Um, I guess to briefly mention how I got into research, I actually took a gap year after high school where I was teaching underserved high schoolers agriculture. Um, and that got me curious about the science of agriculture and, botany, plant science. And then that has a bunch of other little bits of science within it, like agriculture is a bit of chemistry, biology, physics. And so I didn't know anything very well, but I was really curious about those sorts of things. Whereas in high school, I wasn't a good science student and didn't really know what research even was. And then when I got to Rutgers as a freshman, I went to this sort of Friday seminar thing where any student who signed up could go. And there was this interesting presentation about food security given by three professors. And then I talked to one of them who's a plant science professor about his work and my background in agriculture, asked some questions just because I was genuinely curious. Enough of that, started working in his lab. And that's how I spent my first 2 years of undergrad. And then Brinda and I both want to go to med school and do research that's relevant to medicine. And so towards the end of my second year, where I am now, I started thinking about maybe doing research that's more medical. I knew that I really enjoyed doing research, and I worked with 4 different PhD students. And when all 3 of them graduated almost all in one go in the span of a few months. And so it was a really good time to see if I could transition and see if I also enjoyed the research in a different lab, doing something more biomedical. And so that's how I ended up at a liver disease research lab at the Center for Advanced Biotechnology and Medicine, where I met Brinda and Mariam.
Thank you for sharing your getting into research journeys, guys. I also got rejected the first time I applied to the program. I didn't email professors. And then I applied to the same lab again. And we had three chances. Like we were allowed to apply to 3 labs. And I was just only applying to one because I was only interested in that one. And there was hundreds of labs with different research projects, but I was only interested in the one that I'm in right now. And the first one I got rejected, and the second one I thankfully got accepted because I emailed the professor while applying, saying, "Oh, I read your paper and I'm interested in what you're doing," basically. So that's another thing. You can both apply to the program and email them about specific things you're interested in their lab. And now that we talked about how to be involved in research, after you get involved in research, what are some good skills you'd need to possess?
I truly think that being on time is one of the most crucial things. I think that goes hand in hand with ambition and passion. You truly cannot be a good scientist if you don't follow, like, just the basic level of professionalism. So just be on time always, but also be ready to learn. It's about your attitude always. It doesn't matter how much you know, but it's like how much you want to know and want to like keep learning. So I feel like taking really good notes come into good play. Always, so my biggest tip is from personal experience. After you're done learning about how to proceed with an experiment, I usually take the notes and then I go home and I try to write it down like I'm replicating the experiment. And I write this down like 2 or 3 times in order to actually establish a good connection and idea of what I'm doing so that the next time my teacher asks me, "Hey, can you do this?" I'm much more better as a scientist, and maybe new or questions will arise during that process, but I'm much more prepared. And I think those are really crucial aspects.
Definitely when you're doing something complicated with a lot of steps. For us in lab, definitely we all have lab notebooks, and then you want to write down everything you did or see how much you remember. And then so I even leave like blank spaces where, oh, I don't remember what percentage of this we used or what the name of this chemical was. And if we don't do that, then one of the biggest issues is our work becomes unreliable. If I do something twice, but then treat my samples a bit differently both times, then I can't really use the comparison anymore. And so being good at research is really being detail oriented and not being afraid to ask questions. And so sort of being very curious about big picture and small picture, but being willing to get into the nitty-gritty detail of whatever it is you're working on. Oh, I put a picture on the slide. I hope this will resonate with some people. I was on vacation from lab for the past week. And so I was in the library the day before, and I was wondering if there were any books that were sort of related to the stuff I was looking at. And so I found this book, which has a cool cover. So I wanted to show it with, this is the molecule we look at. I guess once it's with iron, it's heme. And so heme is really important for any animal that's alive. It becomes hemoglobin, which you need if you want to be alive. And so it's a cool molecule. We don't look at any of the stuff related to hemoglobin, but anyway, sorry. I took this book out from the library to take with me when I went on vacation, so I could read it while on the way. And what's interesting about it to me is it was written in 1987. To the best of my knowledge, none of the authors are still alive. And what was interesting is when I got into the lab and started working this summer, I thought that our field was very small and niche, and that other people weren't really interested in what we were looking at. But because I was sort of curious, oh, well, let me see what other people used to say all these years ago, and sort of wanting to know more about the process, what happened, which I wasn't expecting, is I got to appreciate the field itself more, and I found out that there's over 150 years of work of people who've worked on the foundation of the stuff I'm working on. And while it's not all or even mostly relevant to the work I do, it's given me inspiration for new questions to ask and for things to bring up during the lab meeting, and new ways of looking at the problem we're studying, which is a type of disease, and why things are happening that way. So I don't know, I feel it's pretty narcissistic to say, oh, look at me, I'm such a good researcher. It's more, look, I think it's because I'm sort of curious about these things, and sort of just naturally interested that it makes it fun. If I wasn't reading these things and having these thoughts, it wouldn't be fun. It would be, oh, why am I spending dozens of hours in lab pipetting and doing all of these crazy procedures when I could be out having fun with friends? And so it's because I'm so curious that it's all worth it, basically. If you have anything to add.
I think it's like skills a good researcher possesses, and skills anyone in doing anything possesses, like passion about what you're doing, curiosity, and motivation, and being able to himself in what you're doing and not being like just not getting it into a routine, always adding something new, especially in research. I think I got to work more on that, but like reading papers, reading books on what you're doing, every time I do that, then I'm more interested in actually being in the lab doing things. It makes it more worth it, definitely. I mean, it makes it worth it. If you don't know why you're doing it, there's not a ton of point in doing it crazy procedures. And you already got a little bit into it, but what are you guys working on specifically? What is your research about?
Oh, I think I can go first then. So I put two pictures on top is a plant, and then the bottom is cutouts from the poster I made this summer in my second lab. As I said, I started out in a plant science lab. This plant is actually catnip, which is named catnip because because cats love it, basically. I'm not even gonna try and make any jokes about it. Cats really love this plant, and if you ever see them around catnip, they'll sort of roll around in it, and they get sort of high off it, almost like it's a drug, in the way they behave. And so the main reason for why this happens, as people found out long ago, is that catnip produces a chemical that's not completely unique, but that catnip produces a ton of, more than most other plants, called nephethalactone. And so catnip, the plant, is called Nepeta cateria because it produces nephethalactone. And this chemical is what attracts cats. And the reason for that is that it is probably an evolutionary advantage for cats to roll around in catnip. They survive better than those who don't. And the reason for that is that apparently the chemical nephethalactone can repel mosquitoes and ticks and bedbugs. And so those three creatures can all spread disease. And so the hypothesis around why cats love catnip is that those who rolled around in it were less prone to being attacked by ticks, bedbugs, mosquitoes, and so getting those diseases from them at a much lower rate. And so why does this matter? What's our lab doing with it? Is that lab that I was involved in was working on ways of modifying catnip genetically and chemically and agronomically. So changing the very genes inside of catnip, or the way those genes work, and changing the way we grow catnip, and even using different chemical treatments on catnip, all with the goal of seeing if it could be turned into a repellent that humans could use. So maybe we could also use this as a new molecule that can repel ticks, mosquitoes, and bedbugs. There's only one product at the moment that's being used as the gold standard for a repellency for a very long time, which is DEET. And it's not a bad product by any means, but there are various advantages catnip could have. And so that was the point of that lab. And I worked with different students, PhD students, on their projects. So helping them in the greenhouse, helping them in the lab, and learning what they were working on, why they were working on it, and asking a countless amount of questions about why we were doing things and how everything works, because I really didn't know very much. And they all luckily loved having these conversations with me. And so we got to learn a lot from each other and had a really good time at the lab. And so I joined that lab knowing that I wanted to be a doctor. Why join a plant lab? But I knew I wanted to find out what research is and if I like it. And all I needed for that was a good research environment. So knowing the professor is nice and the PhD students are great was all I really needed to say, okay, count me in. And so I was doing catnip research for my first 2 years of undergrad. And then at the bottom is the new lab work I'm doing now. I've been there for about 3 months now. And so this lab looks at a liver disease or a skin and liver disease called erythropoietic protoporphyria, which is a mouthful. And so, as I said before about the book with heme, making heme, which is a molecule in hemoglobin, which is in our blood, and so super important for life. Making heme is an 8-step process. And people over time have gotten these mutations where the normal genes for making heme are messed up. And so there's 8 steps for making heme. Each time one of those genes is messed up, that leads to a disorder that's called a type of porphyria. So porphyria is a family of disorders associated with each of these 8 steps. And then erythropoietic protoporphyria is the very last step of the 8 going wrong. And so that's the disease I look at. And that's the research I've been doing for the past 3 months since joining the lab. And so we're asking the question of erythropoietic porphyria is a disorder that affects everybody's skin. Everyone who has EPP has skin issues. They're very sensitive to light, but only a minority of them have liver issues. And so we're curious about both, we're exploring the liver issues specifically. And within that, we're curious about why there's damage happening in the liver of the people who are affected. We know that people who are exposed to sunlight are experiencing skin issues because they have an accumulation of protoporphyrin 9, PPIX, which you can see in the picture. When you have excess PPIX, you have oxidative damage and light photosensitivity. So you have PPIX in your blood, it's exposed to sunlight, and it reacts with sunlight, causing skin damage. And then it also causes, in some people, liver damage. Why is it a liver disease? Why is it happening in the liver and not somewhere else? Apparently, heme gets made in the liver and in the bone marrow, but it's about 80% is made in the bone marrow, and only about 15 to 20 in the liver. And so why is it specifically affecting the liver and not the bone marrow is a fundamental question we're curious about. And if we answer it, we hope we can learn more about the way the disease causes damage in people. And then maybe that can lead to insights that are helpful to ourselves or to other researchers working on this disorder and understanding, how to prevent it or treat it in a better way. And so I'm sorry for taking so long to explain, but basically we're looking at how liver damage is caused and why it's caused in the liver damage and not in the bone marrow. So I spent my summer producing the picture on the right. I produced a few other pictures, but this is the conclusion picture that I made, where I took liver cells and I arrested them in the cell cycle so they would be in a different stage of life. Basically, our cells, they grow G1, and then they double their DNA, which is S. They synthesize more DNA. Then they grow again G2, and then in M phase, they mitotically split. So M is the splitting phase, and what I did is I used a chemical called mecotazole with PPIX, which is the molecule that's problematic that causes the damage as shown in the photo on the left. So I used a chemical called mecotazole to make liver cells divide a lot more. I made them, or not divide more, I made them arrested, so stuck in the dividing phase. And our hypothesis was maybe bone marrow cells aren't damaged, but the liver is damaged because the bone marrow is dividing much more than the liver is. And so I used this chemical to make the liver cells divide, or not divide, but to arrest them at a stage of division. And then our thought process is that by doing this, we can see if maybe when there's more dividing cells, there's less PPIX, less of the problematic molecule inside the cell. And maybe the way the cells work will be better. They'll not have the way, for example, their garbage disposal works messed up. And so what we found, and this has to be repeated a lot more times, and we need much more conclusive evidence, but we saw that when I added the metronidazole to cells, they were indeed arrested in dividing stage. The concentration of PPIX inside the cells went down. And so our hypothesis is that, and we also looked at proteins for garbage disposal of bad proteins. And these also showed that the system of garbage disposal was sort of working better. And so our hypothesis was that when you have more dividing cells, there's less damage caused to the cells, and that's sort of what we showed. So that's our conclusion figure from the summer, basically trying to give a tiny insight where we keep exploring the story of liver damage. And again, it's only a tiny insight for now, in that I need to repeat it over and over and test a bunch of other things so we can find out exactly what's happening and why it's happening the way it is. And hopefully within some more months, maybe a year, hopefully no more, we'll have a much nicer full story to tell. So that's what I've done as a researcher so far as an undergrad.
Thank you for your explanation. So would you like to talk about yours, Brinda?
Yeah, absolutely. Okay, so the lab that I work at right now, we are studying multiple sclerosis, which is an inflammatory disease. And so what we discovered is these are all from past papers, by the way. We gathered all this evidence in order to come up with our hypothesis, but some of the past papers suggested that, ETX, which is epsilon toxin, and multiple sclerosis have some potential link. And so the way we were able to understand this was one of the studies actually showed that ETX, or the epsilon toxin, can breach the blood-brain barrier, and that causes this process of demyelination. So, we have our regular neurons, and these neurons, think of it as they have this tail, and that tail is basically covered with this like plastic like material, okay? It's not really plastic, but just for simplicity and understanding purposes, think of it like that. And that process, that plastic process that we just talked about, is called myelination. So basically what happens in multiple sclerosis is that the the myelination, or the plastic covering, kind of like breaks apart, and that just basically destroys the protective barrier on these neurons. And so that process is demyelination. And just how I just explained it, it basically damages the entire cells overall. So in the first study, it was concluded that our brain has multiple types of brain cells. However, the only brain cell getting affected by the epsilon toxin is or oligodendrocytes. They did find or note that astrocytes also have some sort of trigger, but it makes sense because astrocytes are typically the cells that aid in restoration, or any type of issues that go in your brain. Astrocytes are the one to solve that problem. So it makes sense for its reactivity to go a little off, but the cell that actually got destroyed was oligodendrocytes, and so that was our focus basically. So after we understood exactly which cell we should focus on, we then read another research paper which basically showed that we have a lot of gut bacteria. Everyone has it. We all have multiple strands. One of the main types, or most common, is Clostridium perfringens. And if you take a look at this image over here, you could see that in the gut microbiome of this mice, Clostridium perfringens exists, or C. perfringens. However, when there's an overproduction of this bacterium, this very common bacterium, it actually releases the epsilon toxin into your bloodstream, which then travels all the way to your brain. And if you take a look at the image on the right, you could see that the above image is a very intact blood-brain barrier, so there's no gaps. It protects everything that's going in and out. And you could see that the oligodendrocyte, or like the brain cell, it looks really healthy and really like put together. However, when you look on the bottom, after the ETX comes through, it actually damages the blood-brain barrier, indicated by that gap you see. And when that happens, the oligodendrocyte becomes punctated, or little tiny pieces, or also known as fragmentation. These are two different processes, but you can see both happening in this region, and that just basically indicates this damage on these cells. And it was noted in a study that that damage is what triggers the pathway for multiple sclerosis. And so this was all demonstrated in mice, by the way. And these were all studies that we read through other papers cited below. However, our lab wanted to take it one step further. We know its effect on mice. but we don't know what its effects are on humans. So we read upon a paper where they had two twins. One of them was a really healthy twin. The other one was affected with MS, or multiple sclerosis. And so what they did was they took a part of their gut and transferred it into two mice. And what they discovered was the healthy mice that got the healthy gut had no issues. However, when you look at the mice that received the gut microbiota from the MS patient, it actually showed MS-like symptoms. And, that's how that connection was further established. So then we took it back to humans. And so our lab basically went through this whole immunocytochemistry process where we treated these plates with these plates of human cells with the ETX toxin, and we wanted to test out two factors. So we wanted to test out time dependency, whether like 2 hours or 6 hours has an impact, or different concentrations. So does 0 nanomolars have more or less of a reaction compared to 100 nanomolars? So we did that with 6 different types of concentrations. And when we initially did it, our results did not show significant impact in majority of the regions. I wish I could display it, but it's a lot of data points in order to show. So here's a light summary of like that explanation. But like I said in the beginning, you should always validate research. You should always look back onto what's going on. So upon just observing without tracing these cells, because that's how we were able to understand the morphology and come up with this data, we just took a picture, like took an idea on what these pictures look like. So we lined all the 0 nanomolars and the 100 nanomolars up, and we observed what is happening. And when we looked at the 0 nanomolars, we can visually see that the oligodendrocyte looked much more fuller, had a lot of like lines going through or paths, versus the other oligodendrocyte in the 100 nanomolar treatment, which had a lot of fragment or punctated pieces. And so that led us to believe that something in our tracing could be a potential error. So we retraced all the cells in order to observe what is actually happening. And upon reanalyzing, it actually showed what our hypothesis initially analyzed, for the most part. And so it's always nice to, again, for like just a side note, it's always nice to always redo and see what the source of error is. Never destroy your data. It's always a learning process, but we were able to identify that this was an error on tracing only because we were able to take a step back and understand what was going on. If that was not the case and our tracings appeared to be as good as they should be, then another hypothesis that we did come up for the not significant results was that maybe there's just no effect, or there's a threshold that needs to be met. So maybe the amount of time we could left the ETX toxin to these human cells was not enough, because we tried 2 hours and 6 hours, but maybe it needs a lot longer. That was another part of the hypothesis. And the last one is another part of threshold, but it goes onto the concentration of that. So we discussed how we used ranges from 0 all the way to 100. And maybe it requires a little bit more. So maybe doing like 200 nanomolars or 500 nanomolars, anything higher, maybe that could have showed other results. But we wanted to test out the most easiest one at the moment, which was to actually analyze what was happening with the traces, to see if there was a problem with the beginning work. And after establishing like a guideline on what's background, what's the actual image, and how to trace it again with proper instructions, we made sure to outline everything so that, these tracing errors don't happen again. But you could see how it drastically changed the data and got us to actually show what our hypothesis initially said. So I think that's really nice. And that's what we're working on right now.
Thank you, Brinda and Or. I want to just like very shortly summarize like what I got out of what you guys were saying.
Yes.
Not about your research specifically, but about like what everyone can get like as an advice. So I think in the beginning you ask a big question of why is like the liver being damaged more than the bone marrow, and you then made a smaller, like more details. You asked a more detailed question. Oh, is it because let's say, is it because more cells are being produced in the bone marrow? If I make any mistakes, just correct me.
Yeah, yeah.
And the liver, and you arrested the liver cells in M phase, and so you ask a question, you ask a deeper question, you manipulate a variable, and then you examine the results, sort of. And in Brinda's case, they looked at, like, while trying to answer a question, they varied concentration, they varied time. So you can't, I guess, like, the point of doing research is asking first good questions and then try coming up with good ways of answering those questions. Now, lastly, these are just my interpretations. I think you made a very nice presentation and people can get whatever they get from it. And now, like, to slowly wrap up, for people who are interested in pre-med and watching this, let's say we gave a lot of advice for people just interested in research. Like, in 1 or 2 sentences, what would you, I know you've made a big, so what would your advice be to those who are going into pre-med, other than research or adding on to research, Brinda and Or?
I can give a bit of advice, I think. I think pre-med, and I guess it depends on what country you're in, because the systems are very different wherever you are. We're based in the US, so I can speak to the US, being that you have to do undergrad and only then you go to med school. And so undergrad is, for most people, a time to really explore a bunch of different things and see what you're interested in. And if that's medicine, then that's awesome. And if it's not, that's awesome too. I think and in pre-med in the US, a big problem is that it's so tough to get into med school. You have to do so much that people get very laser focused on sort of, I have to do all of these things. You know, the National Organization for Med Schools requires like 15 core competencies. So you have to show competency at 15 different things. That's a lot of different categories to sort of show yourself being good at just in the span of like 4 years. And beyond that, you want to be involved in extracurriculars, you want to do well in your courses, et cetera, et cetera. So people get very focused on sort of a checklist of what are the main things I need to do, and how do I set up my schedule so I can do all of these things and do them well. And I think that's a good way of getting those requirements done for sure. But the big issue with doing things that way is that you miss out on exploring and on sort of getting the American college experience, where you're taking different classes because they're interesting. You're talking to people from a variety of fields, and you're joining things just for fun. You're doing things without putting them on your resume. So I think that's a huge challenge in the way the system is built in the US right now. And at the same time, I think that doing things that aren't on any checklist, that don't go anywhere in your resume, is actually just as valuable, if not more, than doing the checklist. And that when you talk to people, if you ever have an interview for med school, when you're writing essays for med school, you want to have stories to tell, and you want to have things that you're about. So for me, something that's been super helpful whenever I introduce myself to anybody is that I can say, hey, look, I actually took a year off before college and taught kids agriculture. And what does that have to do with pre-med? Not a ton, not directly at least. But I was able to pivot that into how I love working in education, and just a bunch of really interesting stories that you get when you work on a farm for a year. And so doing things that way and joining unconventional clubs as an undergrad, I think that actually makes you sort of a nicer applicant and a happier person, which is even more important, obviously. Just doing whatever you want, that's good for you and for others, without concentrating on what the admission officer looking at my application 3 years down the line gonna think of all of this. Because when we do it that way, it's just unhealthy and not fun. And then it doesn't make you look good either. So, you know, might as well have fun while you're at it. Obviously, you have to get your relevant experiences, and I'm sure Brinda can talk more to that. But I think you should do special stuff, things that you'll look back on and say, oh, this is something I would want to tell my grandkids about, not, oh, this is checkbox number 12. So that's my biggest piece of advice.
I agree with Or. I think a really important aspect of pre-med is you should not be defined by the science knowledge you know. Obviously, science is crucial when it comes to pre-med. You're going into it because you have some sort of interest for it, but that doesn't make your character. It all comes down to who you are. And so what Or said is absolutely true. Getting that, clinicals and scientific experience is all important and crucial, but it's also nice to learn a little bit about yourself in order to talk to other people, in order to get connected to other people. And like Aura also said, like about classes, we're so determined to just get through like, biochem, orgo, all these classes that we dread sometimes. But it's also like a little bit more. Like just today, a couple minutes ago, Aura and I were talking about like class on religions, myth, and geography. And I thought that was really fascinating. That's something I really like exploring. So just because you're pre-med doesn't mean you're defined by this scientific community. You're so much more than that. And here's a really like amazing motto with life that I resonate with. And I think that life is like a tasting menu. I know people say jack of all trades, master of none. My parents say it to me all the time. But I truly don't take it like that because I feel like everything in life is offered to you and you should have the chance to explore and deep dive in every single aspect. I think that's really crucial. And it's really important in becoming the person you are because you actually don't know what type of impact you can have on others. Like, say a patient comes in, I was able to talk to someone and ease their nervousness because we had something in common. I would not be able to do that just because I read a textbook or took this ergo class. Like, these are not the only aspects to you. So definitely don't think of yourself as trying to checkbox everything because look, you're gonna read everything, do everything with a passion. You don't have to get every single certification in the world. You don't have to, be the master in every single type of patient technique. Show care, show ambition, show passion towards everything. And I don't know if you know this or not, but basically everything around you will help your pre-med experience. I actually volunteered at a dog shelter, and you'll be like, what does that have to do with like patient care or like helping other people? And look, honestly, not that much. I mean, yeah, volunteering, sure, that can help out a little bit, or consoling dogs, maybe sure, like that teaches me a little bit about empathy and patience. But more to that, it actually taught me organization. It taught me a lot more new skills that I can use in the pre-med field. So if you're worried about applications and exactly how to go about the scientific journey, don't. It's really not as stressful as it seems. I know that we have so many requirements here in America, but like Aura mentioned as well, we're so much more than that, okay? Like we can do a lot more things. You can be a swimming champion for all you know, and that teaches you dedication, time management, and so many rigorous things. And I feel like all that can be applied into pre-med track. So I think that's really nice.
When I say it was a meaningful conversation, I won't feel regretful. Really, it was a meaningful conversation, especially in the end. Um, yes, it was about advice for pre-meds, but I think it's an advice for everyone. Do not just see life with just horse glasses. I don't know if that's an actual idiom, but
Oh,
It
uh,
works.
that's a good one. Tunnel
yeah,
vision. Yeah.
look around you. Look for thing that will make you yourself or get to know yourself while you're at it, while you're young. That was a very, I don't think this was a summary of what you said, but I'm just adding new sentences. Now to wrap up fully, actually kind of going from what you guys also said, deviating from science, how would you guys define art in any order you would like?
I think art is a bit of creativity and passion. I don't think it's just a bunch of colors and trying to, just show a pretty image, but I think it means a lot more than that. Like, I truly think that art is a way of expression, and everything around you is an artistic thing. Whether it be the way you shape your wall interiors or the way you design your notebooks before school starts, I feel like everything around you requires this creative lens, even when it comes to crafting a speech when you're talking in front of people. And I think art is subjective. I don't think it's just one type of thing. So it's really nice because you can see all these interdisciplinary things come together. Like even in science, you have art. Even in mathematics, you have art. All those pretty trigonometry, circles, triangles, all that. So yeah,
That's a very good answer. I think I would agree. I think art is sort of art is what it is to you. And I think it's I complain a lot about how in colleges, for example, we're like, oh, you're a humanities major, you're a science major, oh, you're liberal art, whatever. Um, and I think there isn't a separation to the extent we pretend there is one. With art and science, for example, a lot of what science is an artistic, creative, uh, like sort of interaction and experience that people have with their work and with each other, just like art is on a canvas, which is what most people would call it. And we have so much technical skill in music and in painting, and so much less technical skill in a lot of other more scientific fields. So in that sense, I feel like it's not something I would define for someone. It's whatever it is to you, and I think there's art in whatever we do, like Brinda said.
So you're saying art is not what it is, but how it is, how it's been done, kind of.
Kind
That-
of, yeah, yeah.
That's a nice,
Yeah.
nice way to put it. And another question to you guys is what is a movie that you would recommend to your audience? Totally personal. Like personally, what do you guys like? What movie comes to your mind?
Penguins of Madagascar, #3, I think. I really enjoyed that one as a kid.
Great.
Yeah.
I recently watched Night School on Netflix, and honestly, I found it very humorous. So that's definitely something I recommend.
Mhm. Great. And what is a book that you'd recommend to the audience? And if you have like a reason for it, you can say it. If not,
Slaughterhouse-5, I think. It's it's good for anyone who's, I think, a high schooler or older. I remember reading it in high school, I think my junior year. And it was different from anything I'd read before. It's written by Kurt Vonnegut, who's an American author who's written a lot of it's defined as science fiction, but I wouldn't say it's science fiction as much as it is him talking about himself through the lens of fictional characters, but it's him talking about himself. So Kurt Vonnegut was an American soldier during World War II, and he was taken prisoner by the Germans and kept in a slaughterhouse, Schlachtenhofpunkt in German. And so the book is Slaughterhouse-5, where he was kept. And the book looks at a fictional character who goes through much of the same things and then starts having these experiences where he's sort of seeing himself at different points in time. And what was interesting about it is it was so evident that it's Kurt Vonnegut trying to talk about what happened to him and how he coped with the experience of being held prisoner and of all the death and destruction around him. He was held in a city called Dresden, which I think is now the seventh largest in Germany. And during World War II, the Allied forces bombed it to smithereens. They flattened it, just completely destroyed it. He survived the bombing. He was there when it was bombed. And I actually got to visit Dresden, I think about a year and a half after reading the book. I was backpacking with friends in the Czech Republic, and we took a bus to Dresden. And it was really meaningful and interesting to me to walk around there on a nice summer day and have a good time and nap on a bench in a park and think about what he went through when he was held there. There isn't really any local landmark there that you can see related to him. There's some small building far away from the center of the city, which isn't meaningful by itself. But being there was meaningful to me. And then the fact that the city itself was reconstructed entirely. When you're there now, you can't tell that it was bombed at all, much less than it was completely flattened, that it was a very big city at that. So it was really interesting to me to read the book and then get to visit the city where he was. And I think I went on to read, I think, another book or two of his, and I'm always trying to, I have a big reading list that I never get to reading anymore. But he's very well featured in that list. He writes in a way I have never encountered before, and it's just a very different experience reading the stuff he's written and getting to think about it and getting to visit where he was. I think made it all that more meaningful.
I guess it's a good example of self-expression in art and what he did. And also, I wasn't expecting this big of a story.
Yeah, sorry,
You actually
went down and on.
experienced the book in a sense. And Brinda?
I personally love A Thousand Splendid Suns, I think by Khaled, or I'm not really sure how to properly
Yes,
pronounce it.
I
So
don't know either, but I know the book.
it's an Afghanistan story, so I really loved it because of like the writing style mainly. Obviously, the storyline is something I will touch upon. I loved it truly. But I really love the way he kind of phrases everything, and it actually makes you feel like you're there. Some of the emotional scenes actually do tend to make me cry. I'm not a sensitive person, or I don't think I am. So when it actually it comes to a point where a book is actually making me cry, I definitely think the writer did a really good job expressing what he needed to. And I think the emotions are really strong there. I think it shows you not only that there are hardships, but at the end of every hardship, you'll always have hope and light. And I think that moral is really strong to hold on to. So I really enjoyed it.
Great. No, I normally shouldn't be answering this question, but I just want to. So mine is Lord of the Rings, and I like it because it's a story of a journey, which we're all in. So my last question before finishing it, very easy one word question. What is your favorite color?
Navy blue has to be.
We see it on your ring.
Uh, Mine is just blue. I never picked the film. Um,
You guys
maybe,
are
yeah,
proving.
dark blue. I don't know if dark blue is an official color.
You guys both represent your color interests on you. Well, it was very nice talking with you guys. I'm so happy. I felt it once again. It was a very nice decision to invite you guys here. I learned a lot. If people who will watch it, I think will learn a lot. And I'd like to give the stage to Husna for wrapping up.
Honestly, this was such an amazing discussion. Like, thank you, Or, Brinda, and Mariam for joining us today and like sharing your experience as a researcher and like clarifying for others in the audience like what research is and how to become one. Honestly, I'm sitting here, I'm like grappling on the seat. I'm like, dang, this topic is just so good. I couldn't get enough of it. I can listen to y'all like for hours and definitely want to invite y'all again. Also thank you to everyone who tuned in on this amazing discussion. Like this just shows like we this discussion was so good. I think everyone's gonna love it and like they need to share it with others. More people need to learn about this. But yeah. If there's anyone else in the audience who is a student or an expert who's passionate about what they do and you guys want to share your knowledge and expertise through eTalks, you can contact us at argontalks@gmail.com. And see you in our next episode, hopefully. Thank you again for joining us, everyone. Brinda, Or, and Mariam. This was amazing overall. See you again soon. Thank you.
Thank you for having us.
Thank you for having me.